All right, hello. In this video, I outline A3.2, which is about classification and cladistics. So classification, classifying all the diversity of life, has been a really, or was a really foundational stage in the development of biological knowledge. There's a huge diversity of life, obviously, and before we can try to understand it, we need to organize it to make sense of it. It's sort of like anything. If you're trying to study something, you first organize your notes. And so there's a long tradition of classification in biology. And this section is about looking at that tradition, looking at some of the traditional methods, so how things were classified in the past, and then looking at how that has changed over time and what the modern methods are, so what we call cladistic methods. Those are the modern methods of classification. So why do we need to classify things? I already suggested that in the little intro there. There's a huge diversity of life, and to study it, we need to first organize it and make sense of it. So by organizing living things into different groups, that makes sense, we can then study them further in an organized way. On the next slide, I think I have a video embedded which introduces the concept of classification. It's about 10 minutes, so I encourage you to watch that now. But if you'd rather finish this first and watch it later, you could sort of jump forward on that part as well. Sorry, it's not working. I'll share a link to the video. It's just a YouTube video. It's about 10 minutes. You should check it out before continuing with this video. Thank you. Please pause here, watch the other video, then come back to this one. All right, I hope that that was a nice introduction, and it sort of showed how classification has changed over time and how we're classifying things today based on evolutionary relationships. And I liked a couple things in that video. I liked the rabbit sort of analogy there, where they had the population of rabbits and showed how they speciated, and they mapped that onto a tree diagram. So it gives you an idea of how the diversity of life came to exist through evolution and speciation, and how we're trying to see how living things are related. The flower, the Rafflesia, that big flowering plant example, was also a good one that showed how traditional methods of classification based on how things look have produced errors, if we call it an error, if it doesn't match like their evolutionary relationship. So those flowers have been reclassified using genetic data ultimately. Okay, so the traditional way of classifying was by using a hierarchy, starting with the kingdom and going all the way down to a species. And it was a really useful way of organizing all of the life, the diversity of life on Earth. And to be honest, we still use it because we're used to it and we understand it. However, it is the way we've classified things based on their appearance is somewhat arbitrary. We'll talk about that in a little more detail in a minute. And so we're shifting to an approach based on evolutionary relationships. This shift is, you could call it a paradigm shift. So if you're looking for examples of paradigm shifts for TOK, this is one. So the traditional way looks like this. We can put all life into one of three domains: bacteria, eukarya, archaea. And then within each domain, we have kingdoms. Today we generally talk about 6 kingdoms: animals, plants, fungi, protista, Archaebacteria and Eubacteria. And 4 of those kingdoms fit within Eukarya. And the other 2 kingdoms, there's Eubacteria is within bacteria and Archaebacteria is within Archaea. And then within each kingdom, we have several phyla. So one phyla is called a phylum. And then we get progressively more specific. So within each phylum, we have several classes. Within each class, we have several orders. Within each order, several families. Within each family, several genera, or singular genus. And then within each genus, several species, which is the most specific level in this hierarchy. So it's a nested system. Now, it's often arbitrary. Where the arbitrary part comes in is how do we decide if a certain level should be a genus versus a family versus an order, and so on. Generally, if things are put in different, for example, domains, there's a lot of differences between them. Whereas if they're put in different, just in different genera, but within the same family, then there would be fewer differences. They would be more similar. So as you go from the bottom in this diagram to the top, the differences between organisms within those groups would become less. So the differences between 2 different species within the same genus would be less than the difference between 2 different phyla within the same kingdom. But how do you decide if the differences are small enough, if we should jump down to a more specific, or I guess jump up to a more specific level? That's sort of arbitrary. There's no real way to do that. And so this system has limitations, even though it has been very, very useful and we still use it today. One little vocab thing, a taxon is a group. So domain, phylum, class, order, and so on. Those are all taxa. Taxa is just plural for taxon. And that's what that word means. So it's difficult to classify this way, and there's often isn't a reason for doing things. So what do we do today? What's the goal today? The ideal system is based on evolutionary relationships. So all living things today evolved from the same common ancestor about 3.5 to 4 billion years ago. And then over time, living things have evolved and gone extinct and speciated and branched and branched and branched, leading to the diversity of life that we have today. So the goal today in general is to classify things based on those relationships. So to put organisms together in the same groups that are more closely related, and to work backwards that way. So we try to find organisms that share a common ancestor, look at how far back that common ancestor was, and we create groups in that way. And then the groups have meaning. They're not arbitrary, right? They have meaning based on evolutionary relationships. When we classify that way, it's quite useful because we can then predict what other organisms in the same group would be like, because all organisms in that group will have inherited characteristics from their common ancestors. So we can predict that any organism within that group would have those characteristics. So here's an example of a classification based on evolutionary relationships. This is the classification of all mammals. And you can see there's, at the ends there of the tree, the tips, there's what, 1, 2, 3, 4, 5, 6 large groups. And then it could be divided further. I guess there's three bigger groups. There's the monotremes, the marsupials, and the eutherians. And then it gets divided up even further. So monotremes are all going to share certain characteristics. Marsupials are all going to share certain characteristics that their common ancestor had. And eutherians are all going to share certain characteristics that the common ancestor had. Each of those groups is called a clade. So clade is what we refer to these groups as. Okay. And so how do we decide what to put together in a clade? Because we don't necessarily know who's related to who. We only have the organisms that exist today. We have fossils of organisms that existed in the past, but we don't have a complete record and we can't travel back in time. So we have to base our decisions based on what we see today. Now, traditionally, that's been done based on morphological traits. Those are like structures that we can see in organisms. And that often works, but like the example in that video with the flower, the Rafflesia flower, it sometimes leads to mistakes. So a better way is to use genetic data. So base sequences of genes is very good. We have lots of genetic data. Or sometimes we use amino acid sequences of proteins, which of course are the results of base sequences of genes. Either one of those have been used for modern classification. So getting into the details of how we talk about classification in a modern way. So clade. You should know what a clade is. So here's a tree diagram. Each of those tree diagrams is identical, if you look at them carefully, showing the relationship between what? 1, 2, 3, 4, 7 different species, A through G, or 7 different taxa. Might not be species, it might be a larger group. 7 different taxa. And we can see different parts of those circled. So which one of those would you call a clade? Can you guess which one? The answer is a monophyletic group. Now you do not need to know that vocabulary, but you should be able to define and identify a clade in a diagram, a tree diagram like this. And so A, B, C, D, E, F, and G are what's here today. And moving to the left of that is what was there in the past. So any group of organisms that includes the common ancestor of those organisms and all of its descendants, technically living and extinct, would be a clade. So what's labeled there as a monophyletic group, it includes groups A, B, and C, and A, B, and C's common ancestor, and everything that came between, right? So that's what a clade is. If it's missing the common ancestor, or if it's missing a descendant, it's technically not a clade. And so the goal of cladistics is to identify these clades. And we know then that all organisms within a clade are related to each other, will share certain characteristics that they inherited from their common ancestor. Again, we can use morphological characteristics, what we see, structures to do this, and that's what was traditionally used. But much better than that is base sequences of genes or amino acids, because we have way more data. It's way more accurate to use genetic data than it is to use morphological data. So that's what's mostly used today to create these tree diagrams. How do we know how long ago a common ancestor existed? Can we put time on these tree diagrams? The answer is yes, we can. We can estimate. It's a rough estimate, but we can use something called a molecular clock. So just like a clock tells time because we know the rate at which the hands go around the clock, right? It's a consistent rate. We know that the hours, minutes, and seconds change at a constant rate, so we can, because of that, we can know how long ago things happened. Molecular clocks are based on the same assumption, but instead of looking at hours, minutes, and seconds, and so on, we're looking at genetic differences between organisms that are here today. And we're assuming that the more differences, the longer ago the common ancestor was. And that's based on the assumption that changes occur at a relatively constant rate. And so if you have a change happening like let's say it happens every 100 years, just for the sake of argument, and you have 5 differences between 2 groups that you're looking at today, then you might say, okay, well, 5 differences and there's a change every 100 years, so maybe the ancestor was about 500 years ago. That's when they existed. Essentially, more differences would suggest that the common ancestor lived further back in time. So what? These are rough estimates using molecular clocks. Better than nothing, but they're rough estimates because mutation rates tend not to be completely constant because they're affected by lots of different factors. For example, generation time. For example, bacteria have a short generation time. They reproduce quickly. Humans have a long generation time. And so the shorter your generation time, the greater your mutation rate could be because there's more opportunities for mutations to arise. Size of population. In a smaller population, smaller populations are more susceptible to fluctuations. It's going to be less predictable. Intensity of selective pressure. So there will be changes because of mutations, but also changes because things are evolving to new environmental conditions. And if there's a big change in the environment, then there might be a lot more pressure, selective pressure for organisms to evolve. And there would be more rapid, you would expect more rapid changes if there was suddenly a big change in the environment. And so the environment's not constant. And so because of that, these changes are not consistent across time. And there's other factors as well. So it's a rough clock. It's a rough way to estimate how closely or distantly related species are that exist today. So like I said, the best way to make these tree diagrams, which we call cladograms, is by using base sequences of genes or amino acid sequences of proteins. And the scientific principle that's always used is to look at multiple possible versions of trees, tree diagrams, cladograms, and then to choose the most likely based on which is most simple. So this is called parsimony analysis, and it's a common way to do science. We assume that the simplest explanation is most likely. Okay, that's what parsimony is. And so I want to go through a few examples of how you actually would go about building cladograms using really like simplified models. So imagine you have 3 species, and these are obviously birds. Birds again. Sorry, how to know. So we got 3 species of birds, and they're a little bit different there, and we want to know how they're related to each other. What does the tree, the cladogram, look like for these 3 species? So with 3 species, there are 3 possibilities. This was discussed in the video we watched. And these are the 3 possibilities. Look at those briefly. You can see that they're different. Yes, the shape of the trees are the same, but you can see the way the 3 species are organized is different. So either 1 and 2 share the more recent common ancestor, or 1 and 3, or 3 and 2. So how do we decide which one of those trees is right? Well, we can look at molecular data. Now this is a very, very simplified set of molecular data, looking at genes, or portion, a very small portion of a gene for 3 species, and a species that we know is very, very distantly related to those 3. We'll call that the ancestral species, and we're looking at its sequence. And we're looking at the nitrogenous base at 4 positions in a particular gene. Now, of course, when scientists are really doing this, they're looking at multiple positions on multiple genes. More data is always better. This is just for the purposes of explaining the concept. And so you can see some species have the same nitrogenous base at a given site or position in a gene, and some vary. And so we can use that to choose the most simple explanation and identify which tree is most likely. So this is how you would go about doing it. So these are the three hypotheses, the 3 possible trees. Now what's marked there in purple is what would have had to have changed at site 1 for that tree to be true. So if you look at the tree on your left, not that way, this one, that one, you can see at site 1, if the ancestor was A, had the adenine nitrogenous base at site 1, then that would have had to have changed to a C, okay, before 1 and 2 evolved. And so it changed to C at that point in time there, and then therefore 1 and 2 are C. And 3, species 3, never would have changed, so it just stayed as A. Does that make sense? Look at the other ones. So for the one on the left, you can explain the tree, what we're seeing at site 1, just talking about site 1 in this gene. We can explain the tree with 1 change. Okay. For the second tree there, you need to have 2 changes because species 1 and 2 are separate there. So we would have had to have A change to C twice at site 1. And same with the third one. So it's looking like, based on site 1 alone, the tree to your left is the most likely because it's the simplest. It only needs 1 change to happen, 1 mutation to occur. But of course, that's just looking at 1 site, like 1 position in the gene. We should look at more sites. So let's look at site 2 and 3 and 4. And so you can see here, adding in sites 2 and 3 and 4, for the tree on your left, you can explain it with 6 events occurring. The other 2 take 7 events, 7 changes. And so therefore, what's most likely? Well, it's the one with the fewest events. This is the concept of parsimony. Okay, the smallest number of sequence changes is most likely, and therefore we choose that one. Now, the argument to choose the one on the left there is pretty weak. 7 and 6 are pretty close, and we're only looking at 4 sites. But this is the idea. Normally, if we're actually doing this, we'd be looking at thousands of sites, maybe more, for many genes. And then it becomes quite clear which is most likely. All right, for practice, I'd like you to try a few examples. So here's some data showing the traits that various taxa either have or don't have. So if there's a 1 in the table, it means they have that trait. If it's a 0, it means they don't have it. So for example, the vertebral column, the backbone, all of them except the lancelet have that. Okay. So based on this data, in this case it's morphological data, but this is the same concept of using parsimony. Can you make a cladogram that is most likely, that would require the fewest number of changes? All right, so I hope you tried that out and maybe you got this. And you can see if you organize the cladogram in this way, each trait only has to evolve once before the common ancestor um, of the taxa that share that trait. So the vertebral column would have evolved first um, before the common ancestor of the lamprey, bass, frog, turtles, and leopards. And the hinged jaws would have evolved next, before the common ancestor of the bass, frogs, turtles, and leopards, and so on. And you can see the kinds of traits that are useful for making these cladograms are traits that are shared by some, but not all the organisms. If it's shared by all, it doesn't really help us decide how they should be organized. If it's a trait that none of them have, that doesn't help. But if it's shared by some of them, but not all of them, we can use those to make these cladograms. All right, here's a very similar example. In this one, I'll just show you a little bit more detailed how you could go about making the cladogram. So you would first organize it like this, like a Venn diagram. And so you start with the characteristics that's shared by all of them, put that in the biggest circle, and then work backwards. So take a look at that, see if that makes sense. And then you just convert your Venn diagram into a cladogram. So it's the same information, but just organized as a cladogram. So that can be a little confusing, but if you look at it carefully, I think it will make sense. All right, my PowerPoint is giving me a little bit of a tough time, and it's asking me to record every slide separately. So if it seems a little bit jumpy, the video, sorry, that's because I'm just having technological difficulties. Here's one more you can try, a little more complicated. This one I don't have an answer. So if you want to know what the cladogram should look like, come and see me during tutorial and let's do it together. But I encourage you to try this. It's good practice. All right, let's go through some of the vocab related to cladograms that you need to know. Whoa, double head. Okay, I'm just going to leave it like that. So this is a cladogram. You can see at the ends we have the various taxa. They're often a given species, but they could be a bigger group. And you can see there are branches, and a branch point is where we would say a lineage diverges. It's a point of speciation. It's when one species became two. Now, obviously, those speciation events happen over long time periods, so it's a little bit misleading. But it gives you an idea of where the common ancestors are. The common ancestors are at those branch points. Sometimes usually a branch point divides into two, but we're not always sure exactly when branching happened. So sometimes those branch points can be what we call unresolved, and they could branch into more than two divisions. Okay, so this is a useful diagram. It's showing the same cladogram in two different ways. So you can draw them in different shapes. Sometimes they're drawn like the one on your left there, with just straight lines. And sometimes they're drawn like the one on your right with bent lines, but sort of all like the lineages become parallel to each other. In any case, it doesn't matter, they're the same. One thing to note is that you can rotate around any node, and it means the same thing. So notice the chimp and human look like they're in different positions on these two trees, but it means the same thing, because all a tree diagram is showing is which taxa share a given common ancestor. So the one on the left and the right both show that chimp and humans share a more recent common ancestor than chimps and humans share with gorillas. They both show exactly the same information. So the terminal nodes are what's at the end, that they're the taxa, often species, that are here today. The nodes represent the common ancestors. of a group of taxa. We don't always know what those ancestors were. They're hypothetical, right? It's not like we necessarily have a fossil of them. We may, but usually not. And the root is the very base of a given tree or cladogram. This is a nice cladogram showing basically animals. And you can see how the traits that hold different clades together are marked out. So why do we think all mammals are together? Well, there's many reasons, but one is because they all produce milk. Why are reptiles and mammals placed together? Because they all have an amniotic egg, which their common ancestor had. And why are amphibians, reptiles, and mammals put together? Because they all have limbs with fingers, digits. You'll note that there's clades within clades within clades, right? So this whole thing is a clade. All animals are a clade, but mammals are also a clade. Reptiles and mammals are a clade. Amphibian reptiles and mammals are a clade, right? Remember, a clade is all the descendants of a common ancestor, including the ancestor. And so as we go further back in time, as we look at ancestors that go further back in time, we get bigger and bigger clades. If we're looking at a clade with a very recent common ancestor, it's smaller, right? There's fewer descendants of that ancestor. Interesting question on this slide, where are the birds? So birds are a group, a taxa, that we commonly use, but they're actually not a real group based on evolutionary relationships. So what does that mean? They're not a clade, because birds do not include all the descendants of the common ancestor of birds. If we include all the descendants of the common ancestor of birds, it would include other reptiles as well. So birds should actually, if we're basing it solely on evolutionary relationships, should be placed with reptiles. Reptiles and birds should be together in one group. We often don't do that because just we're used to calling birds birds. But again, birds are not a clade. Okay, you should be able to discuss how traditional classification doesn't always correspond to evolutionary relationships, so what we're trying to do in modern classification. In the video with the giant flowers, the Rafflesia, something like that, that was an example. Another example that's in the curriculum is the Scrophulariaceae. That's a mouthful. Also called the figworts. And they were put together in a group, and it was later determined that there were several different groups. So they were put together in a group, the Scrophulariaceae, based on their morphology. And then when we looked at the genetics, we found, well, they're not actually a clade. They come from many different clades. So that classification was incorrect. We still often use the term figworts and Scrophulariaceae because of tradition, even though it doesn't match the evolutionary relationships. So coming back to sort of the TOK ideas, this is again about the idea of falsification in science. I think we talked about this before in a different section, but many of the classifications that were based on morphology have been falsified based on genetic data. By the way, you do not need to memorize Scrophulariaceae. If you're discussing an example like this in an exam, you can use a common name like figworts. So why does this problem arise when we classify things based on their morphology? Why do we sometimes get it wrong? The main reason is convergent evolution. Species that live in a similar ecological niche, you should know what that means now, over time will evolve to have similar traits because they evolve to adapt to the same environment, the same niche. So because of convergent evolution, we can get similarities that aren't similarities because they were inherited from a common ancestor. They're similarities because they evolved to similar environments. So we have to be careful about those things. Those are called analogous traits. We actually want to find what are called homologous traits, similarities that exist because they were inherited from a common ancestor. There's the terms homologous and analogous down there. Here's a nice example. Marsupials live in Australia. Placentals live in other parts of the world. They're both mammals. They're both different clades of mammals that evolved into these different groups independently. Now, looking at their morphology alone, you might want to put the flying squirrel and the flying phalanger together, right? You might want to put the groundhog and the wombat together. You might want to put the placental anteater and the marsupial anteater together in the same taxon. Well, that would be incorrect because you would be basing that decision based on morphology, and these, in this case, these similarities are due to convergent evolution to similar niches. So anteaters in Australia and anteaters in other places look like they do because they both eat ants, right? They have evolved to be able to eat ants, so they have similar traits. A mouse in Australia and a mouse elsewhere has evolved to look quite similar because of convergent evolution, not because their common ancestor was like that. Okay, the common ancestor of anteaters in Australia and placental anteaters did not look like an anteater, right? So those evolved independently, and so this is just an example of trying to explain convergent evolution and why it's a problem, or has been a problem, in traditional classification. Now, if we classify using genetics, we don't have this problem as often. Here's another example, the Scrophulariaceae. Okay, so all of those plants, those flowering plants in this image, were once thought to be the same family, the figworts, or Scrophulariaceae. Looking at genetic data, it's been shown very clearly that they are they should be in different families. So just another example of reclassification based on evolutionary relationships using genetic data. Okay, last thing you need to know about. We used to not have a domain level in our classification. It was just kingdom. Kingdom was the broadest level. In 1977, a new taxonomic level above kingdoms was proposed, and that was proposed to divide up what used to be just bacteria into archaea and bacteria, and then we still have the eukarya. And that was based on ribosomal RNA-based sequences, rRNA sequences. So again, it's an example of using more data, genetic data, to reclassify things and to reclassify them in a way that more closely matches their evolutionary relationships. And look at me! I'm the common ancestor of all life. Of course, I'm not. So that common ancestor down there at the root of this cladogram existed about 3.5 to 4 billion years ago. And in 1977, based on rRNA data, it was determined that all life fits into 3 domains that split from each other very early in the evolution of life. And so we have the bacteria and the archaea. They used to just be put together as prokaryotes and weren't recognized as different clades. And we have the eukarya, which includes the animal, the fungi, the plants, and everything that's left over, which we often call protista. By the way, protists are not a clade either. They're just the leftovers. Archaea are a clade, bacteria are a clade, fungi clade, animals clade, land plants clade, protista not. If you have no idea what I'm talking about, please be sure to talk to each other and talk to me about this topic when you get a chance. That's it for this one. Thank you for watching. Bye-bye.